The goal of this experiment is to obtain both behavioral and neural markers of human higher cognitive function. First, we will explain the motivation for this study and introduce some of the behavioral and neural measures. If you want to skip the background and go directly to the methods, you can click on the link. This project is motivated by a convergence of two types of evidence, both of which suggest a trade-off between two broad cognitive domains, a socio-emotional cognitive domain and a spatio-visual cognitive domain. The first source of evidence is biological. As the science of human brain imaging has begun to mature, it has become apparent that tasks which involve emotion and emotion regulation, social reasoning and empathy, and self-related processing activate one network of brain areas, whereas tasks which are more externally focused, such as visual attention and problem-solving tasks, activate a different network. Here you can see a contrast image generated from the main scanner task we use, which compares social reasoning with scientific reasoning. If we flip from the lateral surface of the brain here to the medial or internal surface, then we can see areas in yellow which are more active during social than during scientific reasoning. These brain areas are also engaged when people are explicitly instructed to focus internally on their own experiences. If we flip back to the lateral surface, particularly if we look at the brain more from above, then we can see blue areas in intraparietal sulcus, the frontal eye fields, and lateral frontal cortex. These areas, which are associated with visual attention and working memory, are more active during scientific than during social reasoning. These comparisons show that very different networks of brain areas are involved in spatio-visual and socio-emotional cognition. However, there is an even more intriguing biological observation that has come to light in recent years. It appears that these networks are not just separate, but that they have an antagonistic relationship with one another. In other words, when we activate the spatio-visual network, for instance by engaging in scientific reasoning, then we deactivate or turn off our socio-emotional brain networks. This push-pull relationship between socio-emotional and spatio-visual brain networks can even be seen in the absence of any task, when people are simply at rest and thinking their own thoughts. These antagonistic brain networks are illustrated by the red and green areas shown to the right of this figure. To the left, you can see sped-up video of spontaneous activity in the brain while someone is lying in the scanner with no explicit instruction other than to look at a fixation point. It isn't just in the brain that we see a trade-off or push-pull relationship between socio-emotional and spatio-visual cognition. Psychological research in three areas also suggests a trade-off in performance between these domains. This is seen in research on autism spectrum disorders, research on sex or gender differences, and research on cultural differences. The most significant of these areas is autism research. It has long been recognized that individuals with autism have some very well-developed skills. Psychologists have made progress measuring and characterizing these skills in recent years, and the data suggests that individuals with autism excel at quantitative, analytic, and problem-solving tasks that are visuospatial in nature. For instance, in the embedded figures task, one has to locate a geometric shape embedded within a larger figure. Individuals with autism tend to be better than average on this task. They also tend to have strengths in visual search, scientific reasoning, and on the most widely used spatial test of general intelligence, which is called Raven's matrices. Here, the participant has to select the figure that completes the grid. Some published studies indicate that individuals with autism have difficulties identifying biological motion. We've constructed a test to assess this skill. This will allow us to see if biological motion recognition is related to other skills that some individuals with autism have difficulty with, such as facial emotion recognition. Research on gender differences also suggests strengths in spatio-visual cognition for males versus socio-emotional cognition for females. The mental rotations task shows the largest and most robust sex difference of all validated cognitive tasks. Here, individuals must rotate a three-dimensional geometric figure in their head in order to determine which of the alternatives match the figure to the far left. This explains the motivation for our study and serves to introduce you to a number of the tasks and the principal measures of neural function we use. With this background in hand, now we will move to covering some of the important details and challenges involved in conducting the study.
The first challenge is to recruit participants from across the undergraduate campus. We wanted to ensure that we had a good representation from different majors, as choice of discipline is likely to track differences in spatio-visual and socio-emotional skill. To assist with recruitment, we use Sona Systems' online experiment management system. After being directed to the site by advertisements, participants can create a username and password to log in. They are automatically given a participant ID number by the system, which they will use for all of our studies. The first time they log in, participants fill out a pre-screened questionnaire, which asks a range of demographic and other questions relevant to eligibility in this and other studies. On subsequent logins, participants see the active studies for which they are eligible, as determined by the pre-screen. They can then sign up for a time slot. Once a participant has signed up for a time slot, they receive a confirmation email reminding them of their appointment immediately after they sign up and 24 hours before their scheduled appointment. Researchers also receive the same confirmation and reminder emails so that they get notification when a participant has signed up for a study. Upon arrival at the lab, participants are consented for the behavioral portion of the study. Some time needs to be set aside for this, as the study is long and complex. To keep up motivation, all the tasks that can be objectively scored have performance-related pay associated with them. Participants must also be instructed in this as part of the consent process. The procedure for the behavioral portion of the study involves 2 separate laboratory sessions in which the participants sit at a computer in the lab and run through a variety of tasks. In between the 2 lab sessions, the participants do a number of online tests in their own time. The online tests are largely self-report measures. Since they are the simplest component, we will cover the online tests first. Participants are sent the link to a shared Google document by email. The Google document contains links to online tests, which are run using SurveyMonkey. To decrease the risk of a breach of confidentiality, participants do not use their name to identify their responses to these surveys. Instead, they use their assigned participant number. Problems with adherence often arise with online testing. To encourage participants to fill out a test properly and in sequence, we add catch questions to each survey. At the end of the survey, and only if the catch question is answered properly, then participants are given a completion code. They must record completion codes in the Google document. This document is monitored by experimenters. Once a participant has completed a batch of online tests and filled out the completion codes, then they can proceed to the next step. Our analyses of the data indicate good compliance in our online testing. This is most likely due to the face-to-face contact that occurs in the first and second laboratory testing sessions. The steps above may also help by giving the participant an impression that they are being carefully monitored. It is notable that we have been much less successful in adherence to instructions when we have conducted anonymous online tests. During the laboratory sessions, participants sit at a computer in a cubicle in our laboratory testing room. Our laboratory-based tasks are generated using ePrime2. Participants must click on a number of files in order in a folder labeled by letters of the alphabet. The files run automatically, with participants starting by entering their participant number. Instructions appear at the start of the tasks. Care is taken to make the instructions straightforward, and the more complex tasks start with a training block. In addition, participants are reminded that they can ask the experimenter if anything is unclear. This is our version of the DANVA 2, a task developed by Nowicki and Duke at Emory University. It is one of the best validated measures of socio-emotional skill. Participants identify if the emotion displayed is happy, sad, angry, or frightened. The intuitive physics task is one of our tasks used to determine spatio-visual ability. Simon Baron-Cohen at the University of Cambridge has shown that individuals with high-functioning autism do well on this task. Participants record their progress and their performance scores on a paper checklist they are given at the beginning of the session. Once the participants have completed the second laboratory session, their total scores are tallied and they are told how much compensation they have earned. Once they know their total amount, we give them the opportunity to donate a portion of their earnings to one of 5 local charities. The lab will match up to 5% of the participants' earning, and they are allowed to donate up to 20%. This serves as a behavioral measure of pro-social behavior. About half of our participants have chosen to donate part of their compensation to charity, mostly at around the 5% level. An even more important real-life measure of social behavior are peer reports. Here, people who know the participant report on their social behavior. Participants provide email addresses of the individuals who they want to report on them. We ask for peers from a variety of categories, including friends and parents. We then send out emails asking people to rate the participant on a variety of dimensions, again using SurveyMonkey. These emails use a separate code from the participant number to maintain confidentiality. Our goal has been to recruit a number of people who do just the behavioral portion of the study, as well as a second group who do both the behavioral tasks and one or both scanner studies. Once participants have completed the first session of the behavioral study, they are invited to participate in a scanner study. This part of the study is optional, and a separate sign-up and consent process is involved. After arrival at the scanner and consent, participants go through a safety check. If this is their first scan, participants are often anxious about entering the magnet. While safety is the primary concern, some care needs to be taken to not alarm the participant and to reassure them that there is very little danger involved, provided they pass the safety check and do not bring any loose metallic objects into the room. A metal detector is used to check that participants have emptied their pockets of dangerous items. It is important that the participant is as comfortable as possible on the scanner bed, as any discomfort may cause them to move during the scan. The participant gets headphones to hear over the scanner noise, keypads for response, and a visual input through a projection screen mounted on the head coil. This is viewed through a mirror. After the participant has been positioned in the magnet, communication is achieved through the microphone. A series of scanner sequences is used to collect images. First, a localizer scan is used to make sure the participant's head is placed in the center of the field. This is important for the image quality. The participant may need to be removed and head cushions adjusted to ensure good head positioning. Next, a high-resolution anatomical image called MP-RAGE or T1 weighted scan is collected. The MP-RAGE takes just over 9 minutes. During this time, the participant practices the task. This scan provides structural information which can be used to assess individual difference in gray and white matter. The localizer scan is used to help position the field of view that is recorded for this and subsequent scans. This is often referred to as slice positioning. The functional scan, often referred to as EPI or echo-planar imaging sequence, can detect changes in blood flow in the brain which occur when one brain area is being used more than another. An important issue is that the psychological paradigm, controlled by the computer shown here, is synchronized properly with the scanner sequence, which is controlled by the scanner computer. The scanner sends out pulses each time it collects a new volume image or 3D picture of the brain, and these can be detected by the computer that controls the psychological paradigm. The main scanner paradigm we use consists of 4 conditions. 2 of the conditions involve text and 2 involve video with sound. Half of the stories and videos involve scientific puzzles, and the other half includes social interactions. This paradigm allows us to examine the effect of cognitive domain independently of the modality in which stimuli are presented. Each video or story is shown for 20 seconds, followed by a yes/no question, which participants have 7 seconds to answer. The blocks are sometimes separated by longer periods in which only a fixation point is presented. This allows us to compare task performance with rest. In addition, the task blocks and rest blocks are long enough at 27 seconds for us to look at how the patterns of connectivity between brain areas that we see in the resting state are influenced by task conditions. The second scanner paradigm we are using involves looking at pictures of a variety of stimuli, including pictures of faces of family members and friends, unfamiliar faces of adults and children, animals, cute-looking animals, computers, and houses. Intermixed with both these scanner paradigms, we present participants with pure fixation runs. These allow us to look at patterns of resting connectivity as discussed in the introduction. Once we have collected all of the data from the fMRI scanner, we can begin the analysis. Analysis of brain imaging data involves numerous steps, including corrections for movement and transforming the data in a standard atlas data space. We have developed scripts that automate analyses of large groups of participants, our data processing stream also involves a number of quality checks. It is very easy when analyzing brain imaging data to simply put one's data into the black box of the analysis and then only pay attention to the final product that is produced. However, numerous artifacts can hinder the proper interpretation of data that is treated this way. We prefer to look at the data at numerous points in the processing stream. For instance, here you can see an in-house program that we call a rainbow plot, which is a plot that shows the raw time course of BOLD, or blood oxygen level dependent signal from a region, with the different task conditions shown by different colors in the background. Here, the dotted time course is taken from a visual region in the brain, and we can see that the activity is lower during the rest periods, which are light blue. This allows us to check that the timing of the psychological paradigm, which we take from ePrime data output files, is properly aligned with the scanner data. The graph below the rainbow plot shows movement. This is a participant who moves a lot, and we can see that periods of high movement often correlate with noisy, bold signal. From time to time, we exclude individual runs or even whole participants when the noise due to movement is excessive. Once we confirm good data quality, we use FIDDLE, a Washington University St. Louis program, to create general linear models of the data for each participant. The GLMs allow us to make contrasts between each event and activity in the brain during each event. Once the contrasts are made, we can load them into CARET, another Washington University St. Louis program, to view changes in activity on the cortical surface. Here's the same image we showed you in the introduction, which compares social reasoning with scientific reasoning. In addition to looking at how functional data relates to task performance, we are also using a technique called voxel-based morphometry to examine how differences in gray and white matter volume relate to individual ability in different domains. The functional data we have collected also affords more mathematically complex analyses, which are based on advanced mathematical methods that are currently under development to characterize patterns of connectivity as well as the complexity of activity within a region. This illustrates some of the principal points involved in a large and complex study, from data collection through analysis.
A number of people have contributed to this research. Regina Cesaro, Caitlin Begany, Kevin Barry, Benjamin Cubitt, Rochelle Hudson, Abigail Dawson, Brian Heisel, Caitlin Siebert, and Megan Knorr. My name is Anthony Jack, and we are all based in the Department of Cognitive Science at Case Western Reserve University.
For more information about results from the study, you may wish to visit our website. Thank you for your interest, and goodbye for now.